And did we
We're
lose
ready.
JT? Do we have you?
You do.
Okay, great.
I think I saw Beth starting the recording, so I think we're in good shape. Or somebody starting, Doug. Anyway, thank you and welcome everybody to the February meeting of the ICOC and the Application Review Subcommittee. Maria, will you please call the roll?
Dan Bernal.
Present.
Leandra Clark Harvey.
Present.
Thank you. Anne-Marie Dulege, Isabel Dudon, Elena Flowers, Mark Fisher-Colbury,
Here.
Fred Fisher,
Here.
David Higgins,
Here.
Steve Julesgaard,
Here.
Rich Lahara,
Here.
Dave Martin,
Here.
Chris Miaskowski, Lori Miller-Rogan, Adriana Padilla,
Here.
Joe Panetta,
Here.
Al Rolette,
Here.
Jonathan Thomas,
Here.
Art Torres,
Here.
Carol Watson.
Here.
Thank you. And Larry Goldstein.
Here.
Thank you. We have a quorum.
Thank you, Maria. We have a bit of an abbreviated agenda today. We have one action item, which is consideration of applications submitted in response to clinical trial stage projects, program announcements, CLINS 1, 2, and 3. Going to begin with a presentation by Dr. Sombrano, followed by summaries by Dr. Sombrano. Gil, please.
Okay, thank you, Mr. Chairman. Good morning, board members and public. Let me just share my screen very quickly and get us going. Okay. So I'm going to present to you the recommendations from the grants working group related to our latest cycle for the clinical reviews. And as we always begin each of our meetings with a reminder of our mission statement to keep that in mind as we go on to the business of the organization to accelerate world-class science to deliver transformative regenerative medicine treatments in an equitable manner to a diverse California and world. So this is just a reminder of our clinical stage program offerings. We have 3 different varieties. Today we have an application for a CLIN 2, which is to support the conduct of a clinical trial. This is a reminder of the clinical budget. We have an annual allocation for the fiscal year of $162 million. So the fiscal year runs from July of 21 through June of 22. Under that allocation, the board has approved 53.1 million. There are 12 million on the table today for the application under consideration. If that were approved, that would leave a balance of about 97 million. A reminder of the scientific scoring system that is used by the grants working group to score and assess the merit of these applications. So a score of 1 means that the application has exceptional merit and warrants funding. It may have some minor recommendations and adjustments, but those don't require further assessment by the grants working group. A score of 2 means it needs improvement. Those types of applications typically go back to the applicant for revisions, and then goes to the grants working group again for another reassessment. Those that receive a score of 3 are sufficiently flawed that we don't allow reapplication or resubmission for at least 6 months, meaning they really do have more to work on before they can come back. The review criteria that are utilized in that scoring are the following 5 questions. First, does the project hold the necessary significance and potential for impact, meaning what value does this project offer and is it worth doing? Do they have a sound rationale, a good plan and design? Is it feasible, including having the appropriate resources and appropriate team to conduct the work? And then lastly, does the project address the needs of underserved communities? This is a slide just to show you what the makeup of the grants working group is. This is a new slide, but I thought it would be useful just to make sure everybody understands the composition of the grants working group and where the recommendations come from. So the scientific working group members are typically 15 in a given panel. That's per Prop 71 and Prop 14 statute. So they are disease area experts. They have regulatory expertise, CMC, product development, other technology expertise that compose that panel. And they are the ones that provide the scientific score on all applications. But in addition, the grants working group includes patient advocate and nurse members from the ICOC, who also serve on the grants working group. They are responsible for the DEI evaluation, providing a patient perspective on the overall significance and potential impact of projects, and also provide oversight on the review process itself. And so the DEI score that you will see in the summary, last summary page, is coming from our patient advocate and nurse members. And then lastly, we bring in scientific specialists who are non-voting. So in instances, we will have gaps in knowledge that we want to fill. And so we bring in such specialists to provide their expert assessment to the group. They provide initial but not final scientific scores on these applications. All right, so focusing in on the application under consideration, this is CLIN 2 13259. The title is Phase 1 Study of Autologous CD4-LV FOXP3 in participants with IPEX syndrome. So the therapy is a gene therapy. It is gene-corrected CD4 positive T cells that become regulatory T cells. The indication is IPEX syndrome, which is a monogenic autoimmune disease that affects young adults and children. The goal is to complete a phase 1 clinical trial, and the funds requested are just under 12 million. So I'll give you a little more background on this particular indication. So immune dysregulation, polyendocrinopathy, enteropathy, X-linked syndrome, I won't say that again, or IPEX syndrome, is a rare autoimmune inflammatory disease caused by a FOXP3 mutation that leads to a lack of regulatory T cells. And so this often manifests in children between birth and 1 year of age. It is fatal, as mentioned, and leads to manifestations such as type 1 diabetes, eczema, gastrointestinal disorders. And so it manifests in a lot of autoimmune elements. So the value proposition for this therapy, the current standard of care options currently for these patients is chronic immunosuppression or allogeneic hematopoietic stem cell transplant. The immunosuppression is not curative and it has significant side effects. Stem cell transplant can be curative, it isn't always, and there are insufficient matched donors as well as some significant side effects such as graft versus host disease for these allogeneic transplants. So other curative autologous gene editing therapies are a long-term goal for both for this group and others who are working on this. And the proposed therapy offers a bridging opportunity for IPEX treatment by having an immediate benefit. It is providing something that would certainly be better than immunosuppression, and it keeps the patient in a much better condition and potentially more suitable for stem cell transplant if it's appropriate. So this is a gene therapy project, and it's why it qualifies for CIRM funding. Other projects that are in the CIRM portfolio that we fund, we have one other group that has a TRAN1 award from CIRM, So that's a translational stage, much earlier than the current application. That is also addressing IPEX syndrome. In that award, the awardees are trying to develop a gene-corrected hematopoietic stem cell therapy for transplantation that would correct the FOXP3 gene deficit, and this would ultimately be a curative therapy if successful. But as mentioned, it is still early stage. The current applicant team has received some funding previously. They received a CLIN1 award to do much of the work that led to their proposal to conduct this CLIN2 or clinical trial under the phase 1. So they had 6 milestones, which were all achieved on time and completed. They also had a DISC2 award before that to develop a therapeutic candidate, which is actually different from the one that is being considered under the current application. So those are 2 awards that they have previously had from CIRM. So the recommendation from the grants working group for this proposal is a score of 1, having exceptional merit and warrants funding. There were 13 members that gave this a score of 1. There was 1 member that gave it a score of 2. The DEI score is a 9.5 out of 10, so it's a very high DEI score. The CIRM team recommendation is to fund this application in concurrence with the grants working group recommendation for an award amount of just under 12 million. So, Mr. Chairman.
Thank you very much, Gil. Do we have a motion to approve?
So approved. This is David
So
Anzini.
moved.
Thank you, David. And the second was who? David Martin?
That was not
Second, Mark Fisher-Colbury.
Mark, sorry. Thank you, Mark. Thank you, David, too. Do we have any discussion or comments by the board?
I'll just
Larry?
raise,
Larry has his hand raised, JT.
Larry?
Yes, thank you. You know, I just want to point out that the question sometimes comes up as to why we should pour significant resources into such rare diseases. And part of the answer is that often when we develop a therapy for a rare disease, the technology that is developed is applicable to much more common diseases. And autoimmune disorders in general are a few % of the US population and probably worldwide. So they're relatively common diseases, and they're mostly not curable. The autoimmune diseases are usually treated by immune suppression, and that is actually a really rough path to go. So this sort of therapeutic approach, along with the others for IPEX syndrome, may lead the way to much more common ailments. Thank you, Mr. Chairman.
Thank you, Larry. Dave Martin?
a comment on that just further is that this is X-linked. And so it's relatively straightforward because theoretically there's only one allele that needs to be corrected. The other is inactivated. I probably have to hit both and get lucky to hit one anyway. But it's easier than having one that's homozygous recessive in terms of treatment. So it's a good choice. My second question, though, is that the $12 million seems like it's quite high for a phase 1 study. I presume that this has been looked at carefully by staff and feel that it's justified. That's my question.
Bill?
Yes, we do look at the budget and do a comparison to other similar projects. So this is an autologous therapy with, where the manufacturing costs and the number of patients being treated are aligned with the total budget.
How many patients go?
So this is between 20 and 36 that they are targeting.
Okay, that's a large number, right? They can probably get an approval on that if it works.
Mhm.
Phase 1 study only.
Thank you, Dave. Any other questions, comments by members of the board? Are there any public comments on this motion?
I do not see any hands raised, JT.
There being none, Maria, will you please call the roll?
Sure. Dan Bernal,
Aye.
Leander Clark Harvey,
Aye.
Anne Marie Dulege, Elena Flowers, Mark Fisher Colbury,
Aye.
Fred Fisher,
Aye.
David Higgins,
Yes.
Steve Julesgaard,
Yes.
Rich Lahara,
Yes.
Dave Martin,
Yes.
Christine Miskowski, Lauren Miller Rogan, Adriana Padilla,
Yes.
Joe Panetta,
Yes.
Al Roulette,
Yes.
Jonathan Thomas,
Yes.
Art Torres,
Aye.
Carol Watson.
Yes.
The motion carries.
Thank you, Maria. That concludes today's action items. Do we have any comments from members of the public on any items of interest?
I do not see any hands raised.
Okay, well, as promised, this was a short and concise meeting. Thank you, everybody. A reminder from Maria that our next meeting is a full board meeting on March 24th, I believe.
Good job, JT. It is on March 24th.
There you go. Thank you very much. And we have
He needs
several
to pay for all his gold stars.
several other meetings before then of various subcommittees. So we will see you in the interim and on March 24th at the latest.
And our
So,
CNS workshop today from 11 to 4.
and tomorrow.
And
That's
tomorrow.
right. Yes. So those of you who are able to, and I apologize for my cat getting in the line of vision here. Those of you who are able to join, this is a workshop that has been put together a great deal of work by members of the team over many, many months, and I think will prove to be most interesting. So if you have time and can make yourselves available to link in for any or all, I think you would find it to be very, very interesting and rewarding time. So thank you. And with that,